today I'm doing the last one, which is stem cell policy. I'm going to apologize to anyone who's in my biochemistry class because they're going to feel like they know this because they took an entire semester on the topic. We're going to flip through it, and we will be out of here Hunter's time me by 11 because I've heard several of you are actually going on a field trip, so I'm just going to try to accommodate that. The reason why this is kind of an interesting topic, I'll explain to you as we go through, but in general it's a good science topic, science policy topic that links research with like religion and ethics and a lot of politics that happens and maneuvering that happens. All these different things kind of put a flag, regulation, all kind of link in together in one particular topic. I also found that, you know, on one hand I thought that it would be kind of definitive. You know, at the beginning it started off with one issue and then it's kind of morphed to several other issues as time has progressed. In addition, I find that every time I think that, okay, we've resolved this, everyone's kind of quieted down, we can move on, then some huge, you know, political, as in the US, some huge hiccup happens and everything gets all screwed up again. So it's like once I start thinking, all right, this is going to be a boring topic to work on for my research, then somebody, you know, a lawsuit or political change or other new innovations come out, which changes and reframes kind of the issue. So we're basically going to start off with, some of you know stem cells, some of you don't. So I'm going to give a brief in the bio for stem cell biology so we can kind of understand why this particular cells are interesting and controversial. We're going to talk about all the different things that are happening in US policy, which is the bulk of the presentation because it changes quite a bit over the past 3 decades. Then I'm going to also link it into the religion and international policies that are going on. And then talk about how the policy impacts research, why you should care, and then last is more of the topics that are of interest right now for scientists and people in the area, which is stem cell therapies as well as tourism, which are kind of go hand in hand. So to start with stem cell biology, and please stop, ask questions anywhere along the line if I'm moving too fast for you. There are 3 kind of general qualities you would say that would define something as a stem cell. The first one being that it's what we refer to as unspecialized. This means that it doesn't have a lot of definitive characteristics. You have, for instance, you know, a hair cell, it looks like a hair, it feels like a hair. You have a skin cell, it acts like a shield there. These are ones that have a lot less definitive nature. So it's unspecialized is the word I use. Scientists tend to refer to as undifferentiated. So it hasn't turned into kind of a terminal cell that only either becomes itself or it dies. Which leads to the second one. The cells also can just continually divide indefinitely. So the cells can grow. And then the last one is they can, as well as grow and divide continuously, they can also turn into a specialized cell. What is interesting here is that you don't know what you have until it's gone. Stem cells themselves are measured now by their products in the future than they are by the cell itself. So the cell will either divide, and there's 2 of them. So we knew the first one was a stem cell, or turn into a specialized cell, so now we knew the one before was a stem cell. But we don't have really good markers that definitively call something a stem cell. Each line looks a little different. People have looked for, you know, genes for what would be stemness, and it never came, we were never able to isolate anything in particular that caused these cells to have the particular characteristics they have. When you generally talk about it. So that on one hand, the pool I think is a little too general, too open to be able to define stem cells in that way. They're located in a lot of adult organs and tissues. So you have them in your bone marrow. So if you've heard of bone marrow transplants, those are the stem cells that create your blood system. So all your immune cells that come out.
Mm-hmm. Are animal stem cells and human stem cells the same or no?
Well, they're slightly different because of the different organisms, but yeah, they're the same.
Okay.
The same concept. Mm-hmm.
So is it possible to use an animal stem cell and a human, or is that crossing too many boundaries?
It could be possible to use it, but the issue we have in medicine is when you cross species, that you can pull the diseases that are specific to it into the human. It's like xenoplantation, and there's that risk and fear. So we do like I think we use heart valves from pigs when we can't find a human replacement. But we tend to shy away from that idea a little bit more and try to have something that's more specialized for the human so that we don't have that fear. There's a lot more screening that's involved when you move from one animal to human. All right, we have them in a bunch of different organs and tissues. We also find them in umbilical cord. So I don't know if you've ever heard of the umbilical cord banking system that they have for stem cells. This is left over after you have a baby. They collect it, they pull out the plasma there and collect it for stem cells that come from the umbilical cord from the child. And then we also can find them from the embryos, and that's actually where most of the controversy tends to surround. And I'll describe that here. So the embryos that we look at are blastocysts. These are around 5 days after fertilization, and it's before it's being implanted into a womb. So this is about the time point when, if you did in vitro fertilization, IVF, where you would implant it back into the woman to try to get it to implant and have a baby. So you'll see it has an interesting characteristic. It's a circle, and then it also has a clump of cells right here. The clump of cells is what we're interested in. The circle part is what's going to become the placenta, and the clump of cells is actually going to become the human baby. So they pull out these cells, and when they do, it dissociates the rest of it. So in the end, actually destroys the embryo. But by their very nature and definition, we know they turn into an embryo. So we know they have the potential to become every cell in the human body, right? Because in the end, it actually becomes a human child. So that's why they have a lot of potential into it, and that's why scientists are interested in them. This picture here is kind of a three-dimensional picture of what a stem cell looks like. It's a real electron microscope photo of a three-dimensional with these are little nanotubules that are associated with it to give it a little depth. There is, though, controversy associated with the fact that you're going from having this embryo blastocyst state and destroying it to create cells. Scientists don't like to say the word destroy. One's even hypothesized, hey, in theory we might be able to clone it all the way back to humans. So it's not really destroying, we're just repurposing it for a small period of time. But in the end, I think that's kind of going around kind of the major issue and kind of sidestepping everything. The reason, oh, yeah.
So as we have heard about the test tube babies and all, so can they just produce these kind of embryos and those things and get those cells out?
Yes. So currently, all the lines that are federally funded involve people who go for IVF. They're going to actually have a baby, and they end up having leftover ones that they don't use. So for instance, maybe they got 15 that were fertilized. They decide to only implant 2 because they don't want to have triplets. They have maybe another one, they use 4, they ended up with 9 that are left over in the freezer, and they can donate those to research, and those are the ones that are used under federal laws. And we'll kind of talk about that specifically in a few steps. But yeah, that is the major source of stem cell lines that are created. So really, you probably are interested in why we care about these, why are scientists excited, besides the fact that you can turn into every cell. There's a lot of different things in basic science that people want to know, is understanding cell division itself, how we kind of replicate, but most specifically how we kind of specialize from this really naive clump of cells that are all identical to these beautiful complex beings that we are in the end, and how early human development occurs. We really don't know that much about it. Personally, I found this out, especially when I was pregnant with my kids, because there's a lot of drugs and other things that it just says, ask your doctor. We don't have anything that says it's okay for the child, and we have nothing that says, we haven't seen anything so far that says this is bad for you during pregnancy. It's kind of a little wishy-washy in this section. There's definitely ones that unfortunately we found out that were bad, but we don't do clinical trials on pregnant women because, this is kind of a particular state, a vulnerable state, and it's just inappropriate. So this will give us a better idea of what's going on in human development in kind of a petri dish. In addition, there's a lot of excitement about using these for drug testing. So what they want to do is take the cells in a culture, turn them into, for instance, maybe cardio cells, and then be able to start screening some of their drugs for toxicity on cells before they actually get to the humans. So when you do a clinical trial, there's several phases. In the beginning, it's just trying to figure out toxicity. It's trying to figure out, what are the doses that we can use? Is it going to hurt the person? Does it kill them, right? You're looking a little bit about how well it works, but a lot to make sure with a really small group of people that this isn't super toxic and that it was a bad idea. It might have worked in the tissue culture, but not in an actual human. And this is one of the steps in between that will also give us some of information about that, and hopefully we'll pull off, before we get to the phase 1 trial, which are incredibly expensive, we'll pull these particular toxic chemicals off before they actually get into humans. There's a lot of paperwork involved in clinical trials, so this is a good thing. And it's obviously cheaper than actually doing a clinical trial. Working with humans is working with a couple cells in a petri dish. But the majority of people are really interested in this concept of regenerative medicine when they talk about stem cells. So it's the idea of taking cells and replacing what's diseased or damaged in your body. So here's kind of a figure we start with. Here's human embryonic stem cells, but as I said, you can turn into anything. So they have them turning into pancreas, which could help if you have diabetes. There's a particular cell that's missing there. Lung, perhaps you have an injury. Blood cells, heart, perhaps you had a heart attack and you have some dead cells in your heart. They can maybe help with that. Neurons, oligodendrocytes for things like Parkinson's and multiple sclerosis. So they all seem to link to things that we don't have a lot of current technologies, drugs or therapies to cure. They have a little bit of stuff we have in medicine that helps alleviate the symptoms of these, but these are a lot of debilitating diseases that there's a lot of interest to see. Can we replace these with our own cells, with somebody else's cells that look like ours, with cells that the immune system will recognize and say that they're okay, and replace particular diseases? And so this is why stem cell research has such a large group of advocates, of people like you guys that go down to Washington, that go to Austin, that protest, that beat down the doors of senators and representatives and push the president to be more permissive, is because they themselves or have family members that are being afflicted with one of these debilitating diseases. I think in science, the biggest advocate groups, and I think Neil kind of hinted this, Neil Lane, when he gave his lecture at the beginning of the semester, the biggest advocate groups are actually for biomedical research because they bring the patients in, a child in, and they come in and look at, if you just funded research in this, I would be better. And we're going to say no. Versus a physicist discussing and atomic energy isn't quite as easy to sell. So any questions so far? So that's kind of the general biology and what you need to know to kind of understand where everybody is and why this is kind of an interesting controversy. So in the United States, all of our policy actually started with the IVF. In 1978, Louise Brown was born. She was the first child born of in vitro fertilization, and this was in the UK. So as you kind of expect, this is actually rather controversial. Everyone thought you were going to actually start having eugenics again. We've had a history in the world, an actual war that was fought that had a lot of this tainted into it, so I think everyone's pretty sensitive about it. In the United States, or the UK where this happened, they spent like a decade and a half having scientists and talking to the public and creating policy. In the United States, we kind of looked at each other, we decided we need to have a little bit of regulation and oversight, and what was created was the Code of Federal Regulations. This is usually what oversees all research that happens and funding that happens in the government. They created Title 45, Public Welfare, Part 46, to oversee any research on embryos. So what it did was establish an ethics advisory board, and they would be responsible for reviewing any grants that anyone had for federal funding that involved embryos. And this happened in '78. So anyone remember who was president then?
Carter.
Carter. Yeah. So Carter was their president. He said this. He put people on the board. They met once to say, "Hey, how are you doing?" And then 1980 happened, and that was. So can you guess why everything started shifting, right? So we started with a Democrat who was pretty liberal, and then we moved into a rather conservative Republican into office, and he just disbanded it. So there was no ethics advisory board, but still this policy on the books. So it said that you had to have somebody review your grant before you could get it, but there's actually no board to review the grant, so obviously there's no research that happens. But you're probably also thinking, we have IVF in the United States, right? And obviously they didn't just kind of, didn't work perfectly the first time. All that is privately funded research that happens in the clinics, and there's no federal oversight over it, pretty much. Now, after a little bit of time had passed, the CDC does require different fertility clinics to turn in their success rates. So they need to know how many they implant, how many pregnancies they have, how many women, things like that, statistics. But in general, all the research on IVF happens in the private sector and not in the public sector in the United States. So this went on for over a decade until 93. So who is now in charge in 93? Clinton, yes. So we switched over to our nice Democrat again. He pulls it off, and there's a small void in power. And while there's a small void right here, it says they could actually do embryo research, perhaps. Obviously, we have to fill that with 94 being an overturn, which the House actually moved from being Democrat for decades to being Republican. Newt Gingrich came in, and they decided to create this, which was the Dickey-Wicker Amendment. So the Dickey-Wicker Amendment is placed on appropriation bills that go through the Department of Health and Human Services, which oversees the National Institutes of Health, which is the one that funds all the biomedical research in the United States that's federally funded. There's a little bit of other fringes, but we'd say like 95% comes from this organization. So they created one that said that no funds could actually be used specifically for the creation of a human embryo. So this would be IVF work, right? Or where embryos had a risk of being discarded or destroyed. So this prohibits all the IVF research using federal funds. And I also wanted to point out they have this elegant, actually, which I'm rather impressed with, definition of what they call an embryo, which comes into play as we move forward, things that are captured here, as well as the fact that this is specifically for the 2009 budget. But it says up here, right, that says it's the 96 is when they first budgeted God on. Every year they attach this verbatim onto the budget that's in some way associated with the Department of Health and Human Services. It might be the appropriation budget for that, or the big, huge omnibus bills that pass where, 10 different agencies get all smashed together, and then they give them money, and they throw this in there as well. So Congress, no matter who's in charge, seems to put this exact same thing. There's a consistency with it, and this was to fill the void because Congress did not like the idea of actually IVF research in the United States. So why this is significant for us, I think you probably already figured out, was because research that we're doing in embryonic stem cells overlaps with this area. And our first controversy actually happened with this cute little lamb here named Dolly. She was created through cloning. In 1996, the paper was published in 97. It was the first time we actually cloned a mammal, and since then we've cloned several others, including cows, and dogs, and cats. But everyone was worried at this point and started panicking that we were actually going to start cloning humans. So there's a big push internationally to start banning this particular type of research. And in the United States, around the world, the United States on the positive side, there's some positive about the fact that our Congress is really slow. Didn't have time to get to it. Because in 98, we had our first human embryonic stem cells were actually created, or discovered. I'll use the word discovered. They're the ones that are linked with all the research. So while we looked at Dolly and even the researchers who did it, they didn't actually think and support this idea of using the technique to create human beings. But when we had all the research that happened in Wisconsin with Thompson's group creating human embryonic stem cells, and as I showed the regenerative medicine aspect of it, there was a lot of biomedical research that we covered here. And so it was this weird balance between saying, no, we don't want this, but we do want this, but they're so closely connected of trying to find legal policy that actually helped with one, but didn't hurt the other, and made sure the reproductive cloning didn't happen.
Yeah, from what I know, the Dixon Amendment stuff is one of the hindrances of this HESES. So why would the Clinton administration supporting this, why would they allow that to hold under Gingrich? Doesn't he have the power to override that, or why would they allow it to?
He would have to reject the entire bill that the Department of Health and Human Services was associated with in order to remove the one little line. They can't just cross off one section of the entire bill. And so it kind of becomes one of those cost-benefit things. I mean, there's a bill, everything looks fine. You're not just doing, I believe they're actually associated with Veterans Administration, so it's like several different areas are all packaged into one appropriations bill. So you can't cross up one little section of it and say, I'll take the rest of it. It's all or nothing. And so I think it's the minimal risk. I mean, yeah, I don't like this particular aspect of it, but again, it was passed through Clinton, it was passed through all of Bush, and it's also being passed through Obama. I mean, it's just kind of managing, we need to fund all of our agencies, we need the government to work, and if this ends up being on there, then it's on there. And we'll talk about how we thought of a workaround, or what we thought was appropriate workaround. So while this was happening, we were with the President Clinton administration. When he wanted to figure out what to do, he actually got together his ethicist, bioethicist. There's a National Bioethics Advisory Commission. The take-home for this is you don't memorize this name, because every administration makes their own. George Bush disbanded this one, and he called it the President's Council of Bioethics, which Obama disbanded and created his own, which sounds kind of like this, but has its own name. But basically, it's a collection of scientists, ethicists, philosophers, policy scholars that come together, that whenever there's something that the president just doesn't know what to do with, that's bioethics-ish related, he just kind of sends it over there and says, you guys analyze it, you guys are the experts, you tell me, give me advice on it. So it's kind of this committee that gets together. So they looked at these issues and they decided that it was inappropriate to clone a human being, but they felt like it was okay to actually fund the human embryonic stem cell research. They felt the government should fund all of it. They felt there was a lot of great potential, and the cost outweighed any risk. But again, they had to actually work with the Dickey-Wicker Amendment, which existed. So you can't just say with an executive order it's all okay, because, well, that'll last for a couple of months, but then when the next budget passes, that actually oversees it, so it'll be this flux back and forth. So Clinton asked his lawyers at the Department of Health and Human Services to kind of read through and figure out what can we do and what can't we do. Does this mean we can't do any embryonic stem cell research with it here? And they decided that there's two separate issues. There's one is destroying the embryo, so that's what happens when you actually create the cells, this line. And then there's the research using those lines. So they said, okay, you can use all the ones after they're created, but you just can't destroy them, so you just can't do the first part, which is the creation of the first line. Does that make sense? It's kind of this weird time point that happens, but you know, the first 5 days are all private funded, but then after that, when you have them all on the tissue culture now, we're all good. You can apply for grants and use them for federal funding. So they went ahead and started all the grants. They said the call, set for reviews. In spring of 2001, what happened here? That happened around 2000, November.
Bush.
Yep, we went from Democrat to Republican. So obviously, every time we switch our administration's tendencies, we switch our policy. So Bush got elected in 2000. He ran on the platform that all this was wrong, that he wasn't going to fund any of it, and the first thing he did was all the review process. So they put the call out, people started writing their grants, but before they even submitted it, Bush was in office and it all got pulled. And he spent some time, he spent 9 months actually looking at the issue. He talked to some ethicists, he talked to some scientists, he talked to the advocates that were in the community before making his official policy, which he released in August of 2001. So what he decided to do was, although he had campaigned saying this is all wrong, we're not going to fund any of it, I think there was a huge pressure, and what's also I think a little interesting about this issue is that it's not necessarily all pro-life. People say the pro-life community must be against embryonic stem cells, but it actually splits a little bit more so. The idea, because most of these cells coming from IVF, that they're left over, they're going to be thrown away anyway, why can't we use them for research? So they still had a strong voice in the Republican Party, and in the end, they pushed that he would put a compromise. And he did more of what you could call a line in the sand. And, you know, at this point, all this bad has happened. I can't do anything about it, but I won't let us fund anything new. So his line in the sand was the date, that August 9th, 2001. He said, if you've already created a line, you've already done it, the bad's over, I can't do anything about it, so we can use those for federal funding. But I'm not going to be involved in the, advocating having new lines created that destroy embryos. So if you create a line tomorrow, it can't be eligible for any federal funding. When he did this announcement, they sent out a press release saying, oh, there are 60 lines, but in actuality there was only 20. It was kind of an overestimation they did based on just kind of random phone calls or talking to scientists of what they may or may not have had in a freezer, whether or not it was viable. In the end, only 21 were actually viable. I also wanted to point out that there's a picture of Bush. The reason why I have this one specifically is he's holding a cute little boy. Has anyone heard of the concept of a snowflake baby? Okay, Krista. Do you remember what it is?
Yeah, the leftover IVF embryos, like Bush suggested that it'd be a great idea for people to adopt those and raise them as their own children.
So he advocated for this adoption program where you would adopt IVF. So now, since Kevin was laughing, why do we think that's not as effective? What's the small little caveat about that? Do you remember?
I do not.
How many babies have been adopted through IVF?
Oh.
Guesstimate.
0.
Like, wasn't it like,
That kid was, so. I think there's a little kid right here.
wasn't it like around 100?
Yeah.
100 or something?
Over like about an 8-year period, there's about 100. And there's estimated about 300,000 IVF frozen embryos that were frozen. So we have the stockpile of hundreds of thousands, and then we have, 100 or two, which, everyone is supportive of it. And that is one of the options you can do, but it's not very popular. So saying that this is what's going to happen to all of them also kind of ignores the fact that most of them are destroyed by being thrown away. So we have this interesting policy where we're really upset about using them for research, and then we just kind of ignore what happens, which is, we're just gonna in the trash can, like it doesn't matter there. It matters when you turn it into research, but it doesn't matter when it just gets tossed because it's not, the parents of the embryo don't have any use for it anymore. I just did a little snapshot of why I think this is significant, but NIH itself, so this is the institution that funds most biomedical research. It has a budget of $30 billion a year. And this is approximately half of what we spend on science in the United States, and it's actually one of the largest funders in the world of biomedical research. So where it goes and where it directs actually leads a lot of other people. Usually, in other cases, it's a leader for ethics, it's a leader for oversight, and at this point by saying we can only use the ones that already exist, it was not involved in any of the discussions directly about how to do lines and how to have ethical informed consent and all these other processes. They were completely out of it because they didn't need it. There wasn't a role for them because they were restricted. And so other, voluntary agencies of scientists had to come up with their own guidelines. In addition, everything that was done here is all federally funded. It has no impact on the private industry. And so that's also why the fact that there is no guidance from NIH is significant, because in other areas of science, biomedical research, NIH was really good about leading, saying this is what's ethical and not, and then the private sector would follow along with it. Because some of them would either link with researchers, so they'd have a scientist at Wisconsin that worked with them, so then therefore they needed to follow guidelines because he was in a university that's associated with NIH, or perhaps even got a little bit of seed money or money for trials from NIH and therefore followed along with it. You're completely removing the government from its traditional role, which is telling us what, guidelines on science and research. We don't work off of having a lot of authorities, agencies. We walk, in science, we tend to have it being a little bit more of a self-government, and that was taking the self-government aspect of it. In addition, there was also confusion because he also advocated some of the states wanted to have more research than what was allowed in the government. You could probably guess based off of, as you've seen, this nice little trend of, a president supportive, a president not supportive. It's kind of linked towards a party. That we ended up with a lot of discrepancies between states. And so this kind of gives you a little snapshot of it. In red are states that have actually banned portion of embryonic stem cell research, and some of them all, the Dakotas, both North and South Dakota, you can't even do what Bush allowed for federal funding. They said no embryonic stem cell research within our state at all. The opposite end, you get California, and they actually passed a $3 billion 10-year initiative to support stem cell research within their state. As almost kind of in opposition to what the president was doing, saying if they're not going to do it, we're going to do it with our state, and everyone's going to come to our state because you can do it here. There's also a lot of states that are gray, including Texas, which really haven't come up with a specific guidelines and policies of what they want to do within the borders. Some of that's bad, but in Texas I actually think that's a positive, because in Texas it seems like every time it's come up in our legislature in the past several sessions, it tends to be in a negative aspect of it. So Corey Kennedy came earlier this semester to talk about the legislature, so they're in session right now. I think still a couple of weeks left. They come every other year and go for about, 100 days. They're in session now. So the first session in 2009 was huge because after Obama came into office, someone tried to replicate the Dickey-Wicker Amendment for the state of Texas. And since all the research had been going on at that point, remember it was 98 when we started seeing it from Wisconsin, that's a full decade going on, he thought, oh, well, no one uses state funds. Well, you know, there's a little money that maybe the state gave for this building. There's definitely a lot of money that went to build some of the buildings that are UT system across the street in the medical center. Maybe a little money went towards salaries. It's a whole mess of confusion trying to sort out if, like, you know, $5 got circulated that was somewhere associated with somebody who did research, especially if it was NIH approved. They had, you know, grants from the government already. They've gone through that ethical guideline. But fortunately, that one didn't pass once they realized they didn't have a handle. The person who put it up was from the College Station area, and he's like, I didn't realize anybody did this research in the state. My answer is that you can actually look on the government website and they'll tell you. 2011, then we had a new change. They just wanted to know who was doing what. So first they asked in between the sessions how hard would it be to do a registry of who's doing human embryonic stem cell research. And then they came up with a plan to actually look at all human stem cell research. The embryo part was apparently dropped in between the two. While embryonic stem cell research is a small portion, stem cell research is actually really big within the state. It's all over the place. There's quite a few therapies, and at that point, that's when all the universities just kind of all dogpiled on this, because this is going to be a complete regulation disaster of trying to keep track of anything that's vaguely related to a stem cell that's going on in the campus. Anything that had a cell line that was in culture, that was cancer, that might be considered a stem cell, having those registration. So fortunately that didn't pass. And in this current session we see a ban on human cloning that covers some general research. We've also found another bill wanting to promote adult stem cell research and have funding associated with it. But not necessarily the stuff that's going on in, like, the medical center that's been proven, more of the stuff that, well, we haven't actually seen a lot of great results of, but it just seems kind of interesting and why not kind of area. So that's incredibly controversial as well. And I don't believe either one of them, because the whole funding issue within the state, we're not spending a lot of money right now. So I'm kind of optimistic that either one would actually pass.
So what's the major difference between the stem cell research and adult stem cell? Because I heard that James Shelley, that took Obama to court, is a researcher in adult stem cell. Mm-hmm. So I was thinking he should be a proponent of stem cell research. So what's the major difference between the adult stem cell and the
What's the difference between the adult stem cell and the embryonic?
Yeah.
The adult stem cells are further along in a process to become more specialized. So you'll find them in an organ, like you'll find them in your bone marrow, for instance. Those cells want to become immune cells. They don't want to become a heart cell. They don't want to become a brain cell. And so you have to do a lot more genetic manipulation in order to turn them into something completely different. Where an embryonic stem cell is supposed to become every cell in the body. So you just have to get it and tweak it and figure out how to kind of coax it to the right pathway. And that's the theory. There's also a lot more, because you start off with a line, you know, definitively, you keep growing them indefinitely. Bone marrow is a great source of stem cells, but other places in your brain are a lot harder to obtain the actual stem cells themselves in enough of a quantity to kind of turn into the amount that you need. But there's still work going on in adult stem cells, and there's also trying to tweak this idea of trying to turn it from blood stem cell to turning it into something else in the body, maybe for your pancreas, to help you with diabetes. There is still kind of an effort to try to see all these different tools and toolkits. What scientists are advocating for is to have the entire toolbox open. As working within an ethical kind of guidelines, having some clear defined rules for what they can and can't do, but having a large toolbox to be able to compare different ideas together and end up with what ends up being best. It might not be best to use embryonic stem cells. It might be best to use another cell source. But then there's a nice control of using those, and they're going to help us understand things in development that will tell us that this other cell line is going to be better. So, oh, did you have a question? Yeah.
How is the stem cell applied into, like, the effective pancreas.
How does it apply to the pancreas?
Yes. Does it transform into a new one, or how does it happen?
Melissa?
It needs to go through several processes. There are about like 5 to 6 steps just to turn it from an embryonic stem cells towards a pancreas cell. And there are several different types of cells in the pancreas. It's not just one type of cell, there are many of them. So in order to make it functional, and that's one the good thing about embryonic stem cells is you get to understand the biology of it, how they can transform. But it's not just one single step. There are many steps.
This single stem cell can actually raise the whole. I think it's another, right?
A group of cells can do, and they can have biochemistry characteristics of the pancreas, and they can produce insulin. Or same thing in the heart, they can actually have movements, and so they are functional, and they can produce another.
Okay. So it's like you are saying that there are other cells apart from stem cells in the pancreas, right?
Yes, because the thing is that stem cell can create a whole person. So they can also create, and they can also create a tissue, they can create an organ. They have that potential. We are understanding how that happens, but they can.
I'm picking on Melissa because she did a presentation yesterday. But yeah, she's saying is there's a collection of cells that are, the immune system has attacked during diabetes, and we're using stem cells to be able to recreate those cell populations. So you're not putting the stem cells in themselves, you're turning them into the cells that were in the population that are missing.
What's the level of differentiation for those cells, to say in an example, pancreas? Or rather, let's say cardiomyocytes. What's the level of differentiation that they actually specialize into the cardiomyocytes from you, I guess?
I think it depends on every one of them. So, okay.
Or maybe I can frame this better. So
we were listening to somebody present the other day in our class, so I'll bring that example into it. They were talking about actually using bone cells, marrow cells, and then injecting them into the heart. And what they found that they weren't necessarily integrating, but they were tracking cells to create a healthier environment to help grow the cells within the heart itself. And what actually I don't think they did, they differentiated a little bit into kind of that area, but I think they actually end up fusing with what's existing there. But it's the fact when they come in there, they release all these different proteins because they were related to the immune system that actually attracts, I think, a lot of cells for healing and repair from the immune system. Does that answer, kind of? No?
There have been any issue whereby maybe a researcher thinks that this stem cell will turn to maybe a cell in the brain, but eventually resulted into another cell that
yes.
not to do.
There is actually a lot of fear and how we use them and use them specifically. For instance, the one we were talking about in the heart, they were looking and they said, okay, and there's also some calcium deposits, like the cells themselves turn into a little bit of bone. A couple bone cells in there. We don't need that in your heart. Really, that is not what you're going for at all, of all places. In addition, there is a worry about tumor formation, especially the kind of, we'll call more younger cells. The embryonic is kind of, we think of as a little younger. It can turn into so many different things. Where if you have the blood stem cells, it can turn into a little fewer. So that's probably also less likely to form a tumor, but still could. But definitely, if you put an embryonic stem cell and you don't know exactly at that naive stage by itself, we have no idea what it turned into. And if you have a couple of those in a mix with ones that you've turned into heart cells, there's a chance that it could form a tumor itself. And then that your cure itself is having a negative impact as much as what you were trying to cure. So trying to make sure to be as precise as possible, and that's kind of some of the issues that are involved in getting this technique and technology. That's why we don't see it as much in the clinic right now. It's just barely tiptoeing into it.
So, stem cell can be the solution for like each and every disease that a person has, which impairs your ability for the cells to perform a specific function?
Maybe, but some of them you have to figure out the biology of why it's happening. One of the interesting things about diabetes is that it's an autoimmune, so it's your immune system attacking the cells that kills them. So you can give new cells and then your immune system comes back on and starts killing them again. So in the end, you actually have to fix the immune system at the same time as helping replenish the cells.
So it is not the similar case with all the other diseases because they are not actually, like consider for a case of psoriasis, where actually the fluid between your bones goes away and your bones start like having
Friction.
on your knee. So can stem cell produce those kind of fluid and actually bring it to proper functioning again or something like that?
I don't know about this specific example, but I do know there is a lot of research going on cartilage and trying to increase cartilage, for instance. But it's all case by case because some arthritis is, again, autoimmune related. So it's your immune system. So you can add them back, but then if they're gone in 6 months, you're kind of in this vicious cycle. So
I think they have been doing that in dogs. I think I heard that with knees and hip dysplasia, doing stem cell therapy for that specific reason. I don't know if it, you know how successful it is because dogs are obviously different from people. But
I mean, we're trying a lot of different things, but there's the problem with where the tissue is what we see, but then there's also the backline, what is actually causing it to. Is it just the cells died? I mean, it's easier to do something like if you have a burn because it came from an external environmental issue, right? You had a fire, you burned yourself, and then we can replace those tissue with new tissue. When you actually start getting into things that are autoimmune, you have to fix not just the issue of what you're missing, but also why it was gone in the first place, why it was destroyed.
Actually, there's one more thing. So, are we using, like you said, the stem cells from the bone marrow for heart also and for pancreas also, or from specific site only we are gathering those stem cells? Or
in therapies right now, the majority of what you're using the bone marrow for is things that are immune system related. There are some clinical trials that's testing around to see if they could use it for other sources. For the heart, that was a clinical trial. And the results were kind of okay. But it was the first stage. They were like, "Yeah, we didn't kill anyone." That was my part of my take home was, it was okay. It wasn't so bad, which in the end is also one of the things that you do in the first stages is making sure you don't do a lot of harm for the people and figuring out what happens when you move the doses around. And they learned a lot in that step. They might even have to cycle through the phase 1 trial again because what they found out was like, oh, we use this really low dose and then we use a medium dose and a super high dose. And then the lowest dose actually had the best efficacy. So they probably should scale back to actually go look around that dose and go maybe a little higher and a little lower around there because the other ones were okay and they had a little bit of efficacy at the smallest dose, which I think they just used almost with that as a control.
Just out of curiosity, do you think you can use it to stop aging since you can replace dead cells and
live forever? I don't know if you can live forever. I'm sure I've had people tell me they've gone to clinics in China and it's been wonderful to help them, with their wrinkles and everything like that. I'm not going to advocate for that. I've never seen anything that says that. I mean, she told me what her age was. I have an aunt the same age. They look the same. So, I don't know. I can't tell, but she thought it was great. But then she was also selling it to not just me, but to all these other people at the table. So she didn't look bad. But yeah, I'm not a big advocate for ones that we don't have any actual data, any scientific data that's behind it. And that's when we'll tiptoe in the last section, which is the tourism and the therapies. So now we're at Obama. So Obama got elected in 2008, and he ran with the platform that he was going to expand embryonic stem cell research. So in March of 2009, he put an executive order doing just that. He took away all of Bush's executive orders and then told NIH and the Department of Health and Human Services to come up with guidelines for research. So the guidelines were released a couple months later, July. They were all ready for them. They knew this was coming in the pipeline. They'd been kind of, creating them on the side, I think, after Obama was elected, just waiting for the opportunity. They were released to the public in about April, so about a month after his executive order, so that everyone could review, and then you have 3 months of people to give, or there's about, I think, 6 weeks for people to give comments, 6 weeks for them to respond to it, and release the new one. What they decided was if you're going to create a new line, they wanted to have the lines themselves derived from embryos that were created for reproductive purposes. So this is IVF, I'm going to have a child, I have leftovers, you can use them for research because I don't want them anymore. We will not use any of these lines, cannot be used for ones created in the lab, and they can't be used with other techniques, only for reproductive purposes. The embryos themselves have to be donated by individuals who've given voluntary written consent. So they also want to make a dissociation between the person that you were involved with when you have IVF to the researcher. They're completely separate. They can't be overlapping. It can't be anyone involved in that clinic who's asking you for it. You can't incentivize people by saying, I'll give you a discount on your IVF if you give some of your away. Nothing like that. They're completely separate. You make a choice, and it's not going to be informed what you feel like your quality and care during your IVF procedure was. That's very important. In addition, we want everyone to understand what they're giving it for. These lines last forever. They're probably going to be sequenced. You have to know that when you're donating, you know, the longevity of this process. In addition, that it has to be a general statement in the sense that you're opening it up for embryonic stem cell research. Some people donate them, but they want it to be real specific. I want it for diabetes. If you have that in your informed consent document, then that means the person doing the research can only do research for diabetes. They also can't be too broad and blanketed, like, sure, everything's fine, because those come across as the patient themselves they're not clear whether or not they completely understood the process. So it's incredibly strict how their guidelines are, and this is just one little bullet that I summarized all of it, but there's actually a series of different things that they bring up, and making sure that this is ethical.
At what stage will the consent be written? Is it before the
While they're donating?
IVF or
No.
after the
It's completely separated. You have your IVF, you realize there's leftover, then they say if you're interested in research, I can actually associate you with somebody else over here. You go and talk to the researcher. And the research, or someone from the research team, as a representative, comes up and talks, this is what we want to use, here's our form. So after the process, you created it for your having a child, and then later, after you've decided that you don't need them anymore, that's when the decision happens.
Don't you think that will be, I'm not sure, I don't know the process of IVF anyway, but I think there can be a shooting inside. Maybe you can start to give the woman a big dose to have more than necessarily fertilized.
And that's why they separate them, so that you can make that decision.
Before rather than after.
Well, the reason it doesn't come at the very beginning is because they don't want to actually do that. That you say you're going to use it for research, so then they give you, there's more incentive to give you a heavier dose and have more. But they have no idea what's going to happen. So the doctor is looking at it for this purpose alone. I'm helping you have a child. So then IVF involves having the woman have a series of injections of hormones that overstimulates, and so that they'll see how many eggs that she can have at once. Usually we only have one, and so they'll try to see if we can produce more. It all depends on the woman, how old you are, and other different factors. How many are released? It could be only like 3. It could be, you know, a dozen. It's all, and then they fertilize them, and then the ones that grow, they throw away the ones that don't grow, and that narrows down the pool. So maybe you were lucky you had a dozen, but that narrows yourself down to 9, and then you decide, okay, well, I'm okay with having twins, so I want to make sure it happens this one time, so we're going to put 3 in, and so you're narrowed down to 6 that are in the freezer, and maybe you're like, oh, well, I want to have a second kid, and you use another 3, so now you're down to 3 that you can actually donate at the end. But they want to give you the process of making all your decisions on your own without thinking about using the research and having somebody saying, oh, no, you don't want to have a second baby, why don't you donate all 6 of them? Or, you know, maybe if we give you extra dose, more things will, and it'll be fine. They want to keep those 2 separate. That makes sense?
Who do the eggs belong to? I mean, do you decide after you're done, I want you to discard those eggs, or I want you to keep them? Who are they? Are they the moms?
Parents.
Or, okay, the parents.
Yeah, and then that also gets slightly controversial. There's like sperm donation and other things like that. But right now, the way the NIH looks at it, it's the parent, so it should be 2 people, right? Because it's a fertilized egg. So between when Bush set his line in the sand and in 2009, a lot of lines actually were created. And so they decided they had to have some oversight in whether or not they could accept those or not. They didn't want to just start everybody all over again creating new lines. So they have a series of requirements for old lines to go that pretty much fall in line with the new line guidelines. But they said, okay, we understand it might not be perfect because